Okay, so hello everybody. This is actually the very first episode of season 2 of our Western Biology Evolution Seminar Series, or WBESS for short. Again, this seminar series is brought to you by our Departmental Research Development Fund here at Western University. And today I have the pleasure to have Matteo Rossi, who is going to speak to us about his, genetic work, the evolution of some genetic work in butterflies. So Matteo, I will let you take away now, and I'll stay in the background while you give your talk, okay? Thank you.
Thank you very much for inviting me, Mateus. I'm Matteo. I'm currently a postdoctoral fellow at the Rockefeller University in the Cronauer lab, working on behavioral neuroscience in ants. But today I'm going to talk about my PhD work that I conducted in the Merrill lab in Munich, and has focused on the genetics of mate preferences in butterflies. All right, so the broader aim of my PhD studies has been that of trying to understand speciation, or in other words, how can new species evolve starting from one? And there's still a lot of unknowns about how this process happens, but it's generally thought that speciation is initiated when populations start adapting to different ecological niches. For example, you could have a population adapting to a more open forest environment versus one adapting to a more dense, closed forest environment. These populations start to diverge, but during the divergence process, it can happen that the two populations come back in contact again. And so for speciation to gene flow to proceed, some reproductive barriers between them must evolve, because otherwise they could, if they mate indiscriminately and mix their genomes, they might recollapse into a single population. And so there are many ways in which these reproductive barriers can emerge, but one that emerges oftentimes is that species actually evolve a preference to mate with conspecifics. And indeed, almost all animal species have mate preferences in that they are usually attracted to conspecifics. For example, individuals that might look like their own or have a particular pheromone. And so I think that this great diversity of mate preferences makes them exciting behavioral phenotypes to try to understand better how behavior evolves, and at the same time, understand better how new species form. And in particular, during my PhD studies, I've been interested in the genetic mechanisms that underlie the evolution of these mate preferences. For example, here you see the males of two closely related butterfly species that are courting their respective conspecific female with this stereotyped hovering flight over the female. And in fact, if you were to have these two species in the same cage, you would see that the males would almost always go to the and court the conspecific female and won't pay much attention to the female of the other species. These changes in mate preference behaviors are underlined by changes in neural and sensory systems, but for innate behavioral differences, eventually these are encoded at the level of the DNA. But we don't know much about the genetic basis of behavioral evolution, and I would argue we know almost nothing of the genetics underlying the evolution of visually guided behaviors, like these behaviors are, as I'll show you later on. And so for my PhD, I've been interested in trying to understand how can these complex behavioral differences be encoded in the DNA of these species. And so at first, for my PhD, I started studying two closely related species that have divergent colorations that you were just seen in the videos that I showed before. One is called Heliconius sydno with this white pattern on the wing, and one is called Heliconius melpomene with this red pattern. The two species started to diverge around 1.5 million years ago, and for the most part, they occupy different ranges of Central and South America. And they also tend to occupy different habitats where you find the white species and a more dense forest habitat, whereas the red species, you find it more at the forest edge, open forest habitat. But still, the two species meet sometimes in an area of sympatry, where they actually still hybridize, although at very low frequencies. And in fact, there must be some strong reproductive barriers between them, because the number of hybrids that we observe is very low. And so some reproductive barriers, yeah, must be there between them. And this is not only in the form of differential habitat use, but more importantly, in the form of assortative mating. And this is particularly clear in males. And this is the behavior that I've been studying in the Merrill lab, and is the behavior that I will talk to you about today. And the way we assay male preference behavior is with a simple mate choice experiment, where we present the male with the females of the two species, and we count how many times the male courted each female type. And from there, we extract a courtship preference score that you see plotted in this graph. And since I'm going to show a few of these graphs during my talk, let me guide you through it. So here on the x-axis, you have the species of the male, and each of these dots correspond to a male individual that has been tested in multiple trials, and the size of the dot is scaled to how many times this particular male courted across the trials. And on the y-axis is this courtship preference score, which is actually the proportion of courtship events directed towards the red female relative to the white female. So if you see values going towards 1, it means that the males courted the red female more, whereas values going towards 0, it means the male courted the white female more. And indeed, we see that the two males have strong preferences for the conspecific females. From some previous experiments, we know that these differences in behavior are largely visually guided, and I will talk more about this later in the talk. Now, to get at the genetics underlying these differences, we can start making hybrids in the lab. And we see that the first generation hybrids and the backcross hybrids to the red parental species are strongly attracted to red butterflies, which shows that the alleles of the red species are dominant, largely dominant for the preference behavior, or in other words, that inheriting one copy of the red species allele is sufficient to make hybrid butterflies strongly attracted to red females. And on the other hand, when we backcross to the white parental species, we see that the phenotype segregates. So we see the whole range of preference scores. And we can actually use these backcross hybrids to study in which part of their genomes having the alleles of the red species or the white species influence male preference. And to give you a better idea of how this works, these backcross hybrids will have inherited different bits of the red and the white species genome that have been shuffled through recombination in the first generation hybrid. And then we can study for associations between having a particular genotype in one location and male preference. For example, we could have 3 hybrid males that are all strongly attracted to red females, and they share a copy of the red species allele at this location, whereas these 3 hybrids that are attracted to the white female instead have are homozygous for the white species allele. So we could say that this genomic location is associated with male preference, although this is a hypothetical example with like 6 individuals, but we can do this for hundreds of them. And this is what Richard Merrill, my PhD advisor, has done a few years ago in what is called QTL mapping. And in case you're not familiar with this type of graphs, on the x-axis, you have the position of genetic markers along the genome, and on the y-axis is the strength of the statistical association between having a particular genotype in the location and male preference behavior. And Richard found 3 regions of the genome that were having the alleles of the red species or the white species significantly influence which female you will be attracted to. And the best supported one and the one with the strongest behavioral effect is located on chromosome 18 that I highlighted down here in red. And we decided for my PhD studies to focus on this region also for two other reasons. One is that in previous experiments, they've shown that this region seems to modulate visually guided mate preferences in particular. And the second reason is that interestingly, this QTL also contains a gene called optics, which the name might be a bit confusing, but this is the gene that is responsible for making the pattern red in Heliconius. And the fact that the color pattern gene and the preference gene are co-localized to the same region, chromosomal region, so they're in tight physical linkage, has some implications for speciation with gene flow that I won't talk about today. But the fact that the color pattern gene is here will become important soon. Still, this is a big region, contains around 200 genes, and the major aim of my PhD has been that of trying to find the actual gene or genes that control for male behavior. And to go about finding these genes, the first thing that we hypothesize is that there might be a change in gene expression between species that determines this difference. For example, you could have a gene that is expressed in different parts of the brain or at different levels between the species that changes how their brain function and eventually the behavior. And we reason that these changes in gene regulation could happen sometime during development, for example, when the circuits are assembled, the neural circuits are assembled, or it could happen, for example, in the adults, perhaps by refining how sensory information flows in a particular neural circuit. And so we collected RNA-seq data from the brain and eyes of the species and their hybrids, and we compared expression levels between butterflies that prefer white butterflies versus those that prefer red butterflies. And here is a short summary of our results in which we found that there are only two genes that are consistently differentially expressed between red preferring and white preferring butterflies. And they are both located in this region of the QTL that is called the QTL peak, which is the region with the strongest statistical association with male preference, or in other words, where we will be more likely to find the genes that influence male preference. But then a change in behavior could also be due to changes in the protein coding regions. And so we studied also for each gene in the QTL region protein coding substitutions between the two species. And we use some programs that can predict whether these changes are likely to affect protein function. And using these programs, we found an additional 3 genes that have protein coding substitution between the two species that look likely to have a functional effect. For a total of 5 candidate genes, that is not too bad, but for a no-model organism to go and try to functionally validate the link between these and behavior we thought would be too challenging. And so actually for most part of my PhD studies, we try to follow other lines of evidence that might tell us which gene in this region is really the gene that is controlling visual mate preference. And so for this, we expanded our analysis to another Heliconius species that is called Heliconius timareta, that is more closely related to the white species called Cyno. And for many characteristics, like for example, habitat use or general brain morphometrics is more similar to Sydno. But as you see here, it shares the same color pattern as the more distantly related Heliconius melpomene. And here I should just take a brief step back, because I haven't told you so far that these butterflies are perhaps more widely known because of their mimicry of warning patterns, where in the same geographical location, you can find even distantly related species that look exactly the same. And this is because Heliconius butterflies are actually toxic to predators. And so by mimicking each other, they can more effectively advertise to predators that they are toxic. And or in other words, the predators predators like birds learn to associate a particular color pattern with something that they should not eat. And so by mimicking each other, these butterflies get a fitness advantage. Okay, now going back to the red species, there is good evidence that this species did not evolve this red coloration through independent mutations, so independent evolutionary events, but Heliconius timareta acquired the alleles to make its pattern red from melpomene in a process that is called adaptive introgression. And so here the idea is that there was something like an ancestral butterfly species that managed to cross to the east side of the Andes, and there it acquired the alleles to make its pattern red by hybridizing with the local melpomene populations. And as we've seen, and otherwise, it went on its own evolutionary path, distinct from melpomene and retaining some characteristics that are more like Sydno, for example, habitat use. And as we've seen before, the gene for making the pattern red is called optics, and is actually contained in the same region that associates with male preference behavior. And so we thought that maybe in similar introgression events, also the alleles for preferring red butterflies were acquired by timareta from melpomene. And so we went on testing this hypothesis. And the first thing we wanted to do is, does then timareta show a similar visual behavior to melpomene? And so together with Alex Hausmann, we did the same choice experiment you have seen before, but this time the red species is this species called timareta. And indeed, here you see the results plotted in the same way as before, and we see that the two males indeed do prefer the respective conspecific females. And then to be more sure that these are visually guided preferences, we did another experiment where this time we present the male with two females of the same species, the white species, where one of the two females has been colored with a control transparent marker pen on the wing, and the other has been colored with a red marker pen to try to mimic the coloration of the other species. And if we do that, we see that the red species becomes strongly attracted to the butterfly that was painted in red, while the white species retains a preference for the control female butterfly, which suggests that the differences that we see up here are largely driven by visual cues. Okay, and then to get the genetics underlying these differences, again, we can make hybrids in the lab. And the first similarity with before we see is that the first generation hybrids prefer red females, which shows that, again, the alleles of the red species look largely dominant for the preference behavior. And then we can backcross to the white species, where again we see that the phenotype segregates, but importantly, those hybrid butterflies that inherited a copy of the red species allele at the same location we have seen before on chromosome 18 have a much stronger preference for red females compared to their brothers that are homozygous for the white species alleles at the same location. And so this shows that probably the genetic basis for visual mate preference might be the same between the two red species. And so we wanted to go and look at a finer scale within this region to see whether we can find some evidence that the two red butterflies share the same genetic sequence at the level of some genes. And so for this, we studied admixture proportions between the two red species. So here we are zooming in the region that associates with male preference. So on the x-axis is the physical position in the region, and then on the y-axis is this admixture proportion value, which is based on the so-called Abba-Baba test. But in a simplified way, all this means is that if you see the y value going up, it's because the two red species share the same allele, and this is different from the white species. Or in other words, they share a similar genetic sequence that is different from the white species. And if we look, for example, at the level of optics, which is the color pattern gene, we see this high peak, an admixture peak, which shows that the two red species have much of the same allele at this location. And in fact, optics is the gene that makes the pattern red in both species. And what got us excited is that there's another peak here on the left that coincides with the location of some of the candidate genes we had identified before that showed protein coding differences or gene expression differences in the brain of the red and the white species. And so we thought that maybe while this admixture peak represents the acquisition for in Timaretta for making its pattern red, this peak here might represent the acquisition of alleles for preferring the red pattern. Still, we wanted to find some other genomic signatures that might tell us that this is might indeed be the preference locus. And so for this, we wanted first to corroborate this admixture proportion results. And so we studied phylogenetic clustering in the region that associates with behavior using genetic variation along the region. And we saw a lot of support for the phylogenetic tree where the two red species cluster together, both at optics and where the putative behavioral genes could be, or in other words, is another way of saying that the two red species share a similar genetic sequence here, and it is different from the white species. We find strong support for a selective sweep at the same location in the species that received the color pattern alleles, which tell us that there was a steep increase in the frequency of a particular allele at this location, which is consistent with adaptive introgression. And finally, we found also elevated levels of genetic differentiation and divergence between the red and the white species at the same location, which suggests that this locus constituted an important reproductive barrier between the species, presumably because different alleles at this location helped the red and the white species remain adapted to their distinct ecological niches. And so altogether, these genomic signatures could be are the ones that we would expect from our preference locus. And so then our next question was, well, do we then see some genetic changes in these locations might be affecting behavior? And so for this, I went back to Central and South America and collect different, I collect RNA-seq data from the brains of different populations of Melpomene, Timaretta, and Sydno, which are actually a bit, within species, are a bit more differentiated than I told you so far, where you could have, for example, slight variations in their color patterns. But the idea behind sampling multiple populations was that we could remove variation between these that is not linked to mate preference, because all these populations prefer red butterflies and these prefer white butterflies. And if we compare the gene expression levels between these populations, we find there is only one gene that is consistently differentially expressed, and this gene is called ribocalcin-1, and it's located exactly under all those signatures for a preferent locus that we have just seen, which we think made it a very strong candidate for encoding mate preference. And so we decided to go and try to functionally validate the link between this gene and behavior. And so for this, I've used CRISPR-Cas9 technology, and I've been I injected many butterfly eggs with a mix of Cas9 protein and guide RNAs that could go at the ribocalcin locus, introduce some double strand breaks in the DNA that could result in a deletion of most of exon 1 and exon 2 of ribocalcin-1, which should therefore make it non-functional. And then we can compare the behavior of those butterflies where the eggs were injected, but that this did not result in a deletion versus those where that have a non-functional copies of ribocalcin-1. And at first, to be sure that the injection procedure did not alter the behavior of the adult butterflies, we coupled in some experiments some individuals that were injected with CRISPR, but this did not result in a deletion, with some that were not injected at all, that we called wild type here. And we paired these two individuals, we put them in a cage with some flowers they can go feed on and a virgin female. And then we can at first study how many times did these males fed when feeding on the flowers. And you see the results here, where on the y-axis is the proportion of feeding by the male that was injected versus the wild type, which means that if you would see values of 1, only the male that was injected when feeding on the butterfly on the flowers, the 0 will mean a contrary, so only the wild type feeds, and values of 0.5 means that the two males fed an equal amount. And this is basically what we see. And importantly, this is the same thing we see for courtship. So again, values of 0.5 will mean that the two males overall courted a similar amount of time in these experiments, which shows us that the injection procedure doesn't seem to have any effect on the behavior of the adult butterflies. But then we see a different result when we paired with the wild types, those individuals that actually had a deletion, a ribocalcin-1, where while these knockout males still feed an equal amount of compared to the wild types, we see a strong reduction in the amount of time that they court conspecific females. And so this shows that a ribocalcin-1 knockout has a specific impairment on courtship behavior that is independent of a broader impairment of motor activity, and we think provides a strong last piece of evidence that suggests that this is indeed the gene that controls visual mate preference in this species. And so to summarize, we have seen that divergence and convergence in visual preference behavior map to this ribocalcin gene, which is not the only genes that is responsible for making these butterflies differentially attracted to the respective conspecific females, but we think is an important one. And then we have seen that the species have shared alleles of this gene, and this has contributed to the evolution of the same visual preference for red butterflies, which we think shows that hybridization in general is an important genetic source for behavioral evolution. Of course, there are still many unanswered questions, and I think a particularly interesting one is that how does ragocalcin affect the functioning of the brain to produce such changes in behavior? Unfortunately, we don't really know. The function of this gene has not been well characterized. It seems to be involved in calcium signaling, but we don't know much more than that. And then unfortunately, also we find this gene a bit all over the place in the brain. So we didn't have many clues as to what it's doing. My guess would be that this gene is changing how visual information is processed between the two species, but I think it would be exciting to test this hypothesis in the future and try to get at the neural mechanisms underlying these mate preference behaviors. And with that, I would like to thank all the people involved in this project, and in particular, Richard Merrill, my PhD advisor, and a lot of the work you have seen today was a close collaboration with Alexander Hausmann. And so a lot of this work is really also his. A lot of other people contributed to different parts of the project. And thank you again, Matteo, for inviting me.
Wow, thank you, Matteo. This has been very insightful. You know, one thing that I really enjoy about about the series itself is that I think the talks so far, each and every talk is very different from each other, and they show that evolutionary biology, you know, can and should be studied under different perspectives, you know. And I think because oftentimes those who know just a little bit about evolution, you know, they have those ideas of one species evolving to the other, replacing each other, so that very linear way of thinking about evolution. And I think, you know, work like yours shows that there are so many, you know, intricate mechanisms that explain speciation and evolution of you know, evolution itself. So yeah, thank you very much. This was very nice. And as I told you before we started recording, you know, my questions are more like high level conceptual questions. And I do have a few notes here. I've made a few notes, so bear with me, I would say. Well, I think, so your work really represents some fundamental concepts that usually are taught in population genetics courses, like undergraduate courses, for instance. And you touched upon very interesting terms. And one of those I was curious about is the actual difference between genetic differentiation and genetic divergence. I do know that those, you know, those are metrics to study population divergence, right? But what is actually FST versus the other one is DXY, right? Which is genetic divergence, right? I mean, now I know those can be very complicated, but, you know, higher level, what do you think?
Yeah, so the genetic differentiation is an estimate of the how much sequence and difference between population, considering like relative to how much they are different within populations.
So we're talking about single nucleotide polymorphisms within that region.
Yes, that is basically the relative, like the difference between two populations, by controlling for how much different they are within the
population. So it's interspecific variation. It's the ratio between interspecific variation over intraspecific variation, right? You're taking into consideration the population diversity within each species.
Yes, exactly. And so, and then genetic divergence instead is just an absolute measure of how many protein coding substitutions there are between the species.
Okay.
And so basically, sometimes, I don't know, you could have like a high DXY value, but it could be that it could be that there's a lot of variation within species that might explain that. So this is a way to control that what you're seeing is more likely a difference between
Between
species.
species, I
Yeah.
see. So having those two values coincide really shows you, it is a strong indication that the difference you are seeing is not just within population of the same species, it is actually between species.
Yes.
Now, when you mentioned you were also looking for phylogenetic support, so the trees you've built there, were they single gene trees or multi-gene trees?
Yeah, so here the schematics, like, it's not really three species, it's actually use five species. And then these trees are built, basically you divide the genome in like windows.
Oh, yes.
And you use like you take windows of like, okay, there are 50 SNPs here between species, and you construct, with that variation, you construct the phylogenetic trees. So it's not really a fixed window size, but it's more like we take windows of 50 SNPs and we build trees starting from those.
Yeah, that's actually really nice, right?
Yeah.
Because again, I think when people think of phylogenetic tree methods, we often think of building a single gene tree or a multi-gene tree or a whole genome tree, right? So we have some I've seen work on super trees in which they do the whole genome. So phylogenetic trees based on entire genomes, but I did not know we could do, like you said, split the genome in windows and run those trees on those windows. Now, my other question for you would be more on the conceptual, in terms of like schools of thoughts, in terms of evolutionary biology, because I think your work, it is very beautiful because it shows us, again, evolution happens not just through the very gradual change of species, right? You talked about adaptive introgression. In this particular case, it seems as though that hybrid zone allowed one gene, it's hard to make generalizations, but like it shows that gene that gives rise to the red coloration has had adaptive value and then spread in the new population through a ancient hybridization event there. And I think, again, especially at the undergraduate level, when students think of evolution, I think they often think about allele frequencies, right? And how populations diverge over time as they accumulate, different frequencies in different alleles. Within that spectrum of, evolution happens mostly through gradual changes of allele frequencies. And in the other end, evolution can be saltatory, perhaps, can happen through, quantum leaps. Where would you place yourself? I mean, I know, one can say, oh, it's both, right? And it depends on the species. But what do you think? Do you think we are seeing more and more cases of evolution happening not only through gradual allele frequency change?
Yeah, I would say like in the last 20 or 30 years, I would say maybe there's been a bit of a change in reconsidering like the genetic source of adaptive variation. I don't know where I stand. I think like it also depends a bit on which timescales we are looking at, and then that these butterflies are, I wouldn't say it's a special group, but we know there's a lot of hybridization going on there, and you have other systems like cichlid fishes.
I was going to say the cichlid,
where you have I think clear examples where hybridization has been very important, and it might even seem that when you have a lot of hybridization, you can have sort of explosions in the diversity of phenotypic traits, because the hybridization reassemble all these genetic variants. Yeah, I don't know. I mean, the previous view is also like still holds true, and then
of course. Oh yeah, absolutely.
there's like a window when species like can hybridize, and at one point speciation is complete, and then you have other processes taking over. But I think it's definitely, there's been a new appreciation of that hybridization is quite important.
Yeah.
Some lineages for like a quick adaptive
A little
evolution.
faster, yeah. No, I agree with you. Yeah, I must say, I want to reinforce this actually. It's good you brought this up because, by no means I'm here, I hate when people come and say like, oh, Darwin was wrong. I think evolution is evolving. It's like evolutionary biology as a field continues to evolve. And this is what happens with science, right? And I think the beauty of the theory of evolution is that the foundation is still there. And, there is a very rich literature proving, the effects of natural selection, the effects of, gradual evolutionary change through allele frequencies. I agree with you. I just like to raise, to bring to our attention that, just like you mentioned, there are other mechanisms like adaptive integration, I would say in plants as well, right? When we're thinking about polyplodization, I think it is clear now that polyplodization is a tour de force, right? In terms of creating new species. So yeah, I think the field, as we are, acquiring new technologies and being able to assess, the genetics of evolution in a much deeper level, and also, in a broader scale, I think these new mechanisms are, we are becoming more familiar with them. But yeah,
Yeah.
I agree with you. I just don't like you. We know those, right? Like,
No,
when certain magazines come and say like, "Oh, Darwin was wrong," it's like it's a little misleading.
No, I completely, yeah, I agree with you. And I be like this project actually, I think like, assuming that hybridization, like it started a bit thanks to the work that has been done on the color pattern genetics. And we thought like, or maybe you can actually use this, genomic signatures to try to map this gene. And so in a way, I would say that was like, it showed that is important, but overall, yeah, I wouldn't say it subverts any.
Oh no, yeah. What's been
for understanding
before.
evolution.
Yeah. But again, I like this idea of showing other mechanisms. It's not just natural. I mean, you didn't touch so much upon natural selection itself, but I think it's the contingency, that's my point. The contingency, like ancient events, they will have effects down the road on evolutionary capabilities and things like that. Yeah. I actually really like when I started this series, that was my hope, to discuss these things, these more higher level things of, natural selection versus non-adaptive mechanisms. What's more important? Or, evolution is gradual or it's saltatory. These are a little controversial things. And we know usually those topics, they tend to pull people apart. I don't subscribe to enough of those polarizing approaches. Now, just out of curiosity to wrap things up. So you mentioned, so this is your PhD work. It's very nice, very beautiful. It was done in the Merrill lab. And in the very beginning, you mentioned where you're doing your postdoc now, and I think you've switched to a new model system. Now you're studying ants, is that right?
Yes. So I feel like, along the way during my PhD, I got more and more interested in, the link actually between the brain and behavior, and not only the genetics part. And so I wanted to go more into the behavioral neuroscience.
Oh, I see.
Hopefully with like an evolutionary approach. But so I switched. Yeah, I'm at the Rockefeller University in the Daniel Cronauer's lab, and I'm studying behavioral neuroscience in ants. So this is a particular type of ants in that it doesn't have queens, and so all the worker females reproduce, and they reproduce asexually, which makes them, you can do some genetics with more ease with them. You don't have to like have a queen. And so it makes it like an exciting system to that has genetic accessibility to study, the evolution of social behavior. And I'm particularly interested in understanding what's happening in the brain of these social insects. Yeah, it was a bit of a change. Eventually, I hope to, bring it all together and study the genetics and the neuroscience of behavioral evolution in the future. But yeah, that's
very cool. Yeah, it's nice. I know the feeling of trying to put together different fields that are somewhat related, but not at the same time. But in your mind, somehow it makes some sense, but it matures, right? Over time, you start new connections emerge in your own mind as you study more these systems independently. Well, with that, I think we can wrap things up. In a second, I will stop the recording for us to finish the episode. But then you and I, we can just chat for, just chat for a minute or so. So yeah, I'll press the stop button now. For those of you who are watching us, thank you for being here. This talk is being recorded now, and it will be the first episode of season 2. So stay tuned. I'll be sending emails and advertising the season 2 again. So yeah. Matteo, thank you very much for being here with us today. Okay.
Thank you very much for inviting me.